receptor ligand binding is describing the interaction between a receptor, which is usually a protein, and a ligand, a small molecule. Uh, in this concept, we're looking at receptors in the terms of receptors being integral membrane proteins on the surface of cells. And these receptors bind to ligands. And so here you see an example of a ligand, and this would be a polar or charged molecule. It could be, right, a formal molecule, could be a peptide like insulin, which is, again, polar, hydrophilic, charged. These items, because they are polar charged, cannot enter into the cell, but they're required to bring about some biologic response within the cell. So, for example, insulin binds to the insulin receptor, and we have a phosphorylation cascade, which is going to cause some responses within the cell. For example, to upregulate insertion of glucose transporters into the cell membrane and to upregulate glycolysis, right? So that we're burning up the glucose. We're also going to be upregulating glycogenesis, right? So we're going to start to package away glucose in the form of glycogen. So we're trying to get those glucose levels down in the blood. And insulin can't enter the cell directly to bring about those changes, so it needs to bind to a surface accessible receptor site. And that's the idea. Now, in this example, we're looking at these receptors as being bound to the surface of the cell, but indeed, this concept of receptor ligand binding applies to soluble receptors, meaning non-membrane bound receptors like myoglobin and even hemoglobin. So if we look at a soluble receptor example, we can consider myoglobin as a receptor and oxygen as a ligand. Now, this principle of receptor ligand binding allows us to determine how well or how tightly a receptor binds to a ligand. And we can describe this also as the affinity of a receptor for a ligand. And as a measure, a numerical measure, we could talk about how tightly a receptor binds a ligand by describing what are called dissociation constants. And so a dissociation constant is actually a thermodynamic constant. And we're going to look at where this is coming from and how we can measure this in the lab. Your homework assignment focuses on you determining or measuring these dissociation constants. And I know it sounds like a lot, but it's not a lot. Just trust me. It's not a lot. Okay. So scientists like to put numbers on things because we don't like to talk in relative terms, like this is better, this is worse, this is strong, this is weak, this is not as strong. So we want to put numbers on these quantities so that we are definitive and we understand exactly, what we're talking about in the system. This idea, right, of receptor ligand binding was first determined by using cells in culture, and it was recognized that you could apply small molecules to cells growing in culture, and these small molecules would bind to the cells. And scientists wanted to go past this and start to define, what exactly is the target? What exactly is the molecular item that's binding to these ligands? And so we start to look at what are these receptor ligand systems. Now, these are non-covalent interactions, so they're reversible, right? And anyway, receptor ligand binding involves a concept here of it's specific, or it has specificity. So a receptor recognizes one ligand or one 3D binding pattern. So there's one spatial pattern for binding of a ligand, and it's all about molecular binding interactions or weak non-covalent interactions between the receptor and the ligand. And I put like a receptor recognizes one ligand, and I wish you could see the air quotes that I'm doing right now. Receptor recognizes one spatial binding pattern, and of course more than one small molecule can present the same 3D binding sites as another ligand. And so one receptor will bind any small molecule that offers that same 3D binding pattern. right? And this is why, well, I mean, if you think about drugs, like small molecule drugs, and you think about drug companies, and you have one drug company that'll add a methyl group to a drug, and they'll say, oh, that's a new molecule. We're going to patent that, and we're going to make millions of dollars because it's got the same binding pattern, but by adding that methyl group, we've changed the half-life in the body, right? So anyway, it's that concept. So the key thing to note here is that the more binding interactions you have between the receptor and the ligand, the more stable the complex, the tighter the binding, the lower, right, the energy of that system. So it's all about binding interactions. The more weak non-covalent binding points of contact you have between the receptor and the ligand, the more stable the complex and the tighter the binding. This will manifest itself numerically as a lower KD. You will have a lower KD. Now, this receptor ligand interaction is also saturatable. So what do we mean by saturatable? There's a finite number of receptors on the cell with a finite number of ligand binding sites. So it's kind of like, the parking lot that's over near the Academic Success Center. There's a certain number of parking spaces, and once they're full, and no more cars can fit in that lot, right? So it's saturatable. Now, we can also look at a system like this, and we can say that we can outcompete binding of one ligand by adding a second ligand. So the first ligand here is the red ligand. And so the red ligand is shown here bound to these cells in culture. If we add a second or a different ligand to the cells, we can outcompete the first ligand. And so we could displace the binding of the first ligand by adding this second ligand. And so this is the concept of, right, competitive binding. And when we look at enzyme kinetics, these same concepts apply. So an enzyme is specific for a substrate. An enzyme is really specific for a pattern of binding contacts on a substrate and positioning of a sessile bond, right? A bond that can be broken in the proper location in the context of catalytic residues on the enzyme. So enzyme substrate interactions are specific. Enzyme substrate interactions are also saturatable, right? There's only a finite number of enzymes in a system with a finite number of active sites. Once they're all filled with substrate, they're all filled with substrate. Can't process anymore. And if we look at enzyme substrate interactions, we could describe things like competitive inhibitors. So we can add small molecules into the system which compete with binding of substrate at the active site of an enzyme. So everything you're learning about receptor ligand interactions is also applicable to enzyme substrate interactions. Let's look at some of these definitions of these thermodynamic concepts here. So if we look at R total is the total amount of receptor, usually a protein in a system. And we can't always know this with 100% certainty experimentally. We can do things experimentally to kind of back determine this. But if you think about, well, how many receptors are present on the surface of a cell, it might not be possible to know that, right? If we think about how much myoglobin is present in a system, that we know. And we can know that because myoglobin has a distinct absorbance band. And you guys saw this in lab, how it has this reddish color, and that, we have the Soret band absorbance at 409 or 417, depending on the iron oxidation state. So for some things we could know R total, but for most of the things we don't know R total. The receptor ligand contact complex. Let's look at the description of RL. This is a bimolecular complex because we're looking at two species that come together. This receptor ligand complex is formed through random diffusion. Random diffusion of the ligand onto the receptor. And this is a very slow process if we think about this experimentally. Um, so it's not like the receptor and the ligand see each other across the test tube and they run towards each other knowingly, right? Oh, receptor, how I've missed you. Oh, ligand, right? That doesn't happen. There is random diffusion or random motion of the receptor and the ligand, and they're both tumbling, right? If you think about a free receptor and a free ligand, they're both tumbling around in solution, and eventually they're going to bump into each other by chance in the correct orientation. And if they do that, then we will have binding occur. So it's a very slow, random process, random diffusion. It's a very slow process. And so the receptor ligand complex is also a non-covalent complex, and I've mentioned this to you already. This binding interaction is reversible. No covalent bonds are broken or formed in making this RL complex. It's all about the weak non-covalent binding interactions here. So what are those weak non-covalent binding interactions? Well, we have hydrogen bonds, we have hydrophobic effect, ion-ion interactions, which is salt bridge formations, ion dipole interactions, and van der Waals forces. So these are the weak non-covalent interactions which are important for making this RL complex. And the more number of binding contacts you have, the more stable the interaction and the lower the KD. Low KD equals good binding. Now, the receptor, also known as R-free. So if you ever just see R, assume it's R-free. This is the free unliganded receptor. Saying unliganded is the fancy way of saying that there's no ligand bound to that receptor. So R total in a system where we've added ligand is going to consist of receptors that are holding ligand, so RL complex, and R-free. Okay, so that's the idea of what is R total in a system. Then we have ligand concentration. And whenever you see just a plain L, assume that it's meant to be L free. This is the amount of free ligand in a system. And so, ligand concentration is typically varied in experimental determinations of KD. We know L total experimentally. Right? We know L total because we control L total. And we can usually assume that L total is approximately equal to L free. And now I want to show you why and how we can make that assumption. So let's look down at the very bottom of the page here. And so we can assume that L total is about the same as L free because R total in a system is usually very, 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 very less than L total, right? And so, right, if you look at the number of receptors on a cell, some receptors are present in a very, very low copy number on the surface of a cell. And it's such a small number that, it's negligible compared to the amount of L total that we add to the system. So let's say that we add 1,000,000 molecules of L to the system, and we bind 50. And we'll say that that 50 is maximum number of ligands that can be bound. Well, what's 50 out of 1,000,000? It's like no change, right? So we can assume that L free in our system is the same as L total because the amount of R total is so small compared to the amount of L total that we're adding to the system. So that's why we can make that assumption. And we control, right? We control L total, right? Because, right, we add. We add ligand to the system experimentally. All right, let's get back the laser pointer here. Now, if we look at Bmax, Bmax is the number of binding sites, and this is experimentally determined. This is not necessarily the same thing as receptor total for a system, right? Not necessarily always receptor total, because Bmax is the number of binding sites total, and understand, right, that some receptors have more than one binding site. So if we look at myoglobin has one binding site for oxygen. Hemoglobin has 4 binding sites for oxygen. And so Bmax and R total can vary depending upon the number of binding sites per receptor. And we can experimentally determine this. The next item that we're going to look at here is what you're going to be calculating for homework, this dissociation constant. A dissociation constant is an equilibrium constant describing the dissociation of a receptor ligand complex. And here's the reaction we're dealing with, right? This is the reaction we're dealing with. So we have the receptor ligand complex, which can break down to give us free receptor and free ligand. And we're really looking at an equilibrium expression. And if you look at a KEQ for this equation, KEQ is always the ratio of products to reactants. And so we have the concentration of the receptor times the concentration of the ligand divided by the RL, right, concentration here. Now, you know, I told you that this is like a thermodynamic, it's a thermodynamic constant, this KD. But buried in there, we have some kinetic constants. So we have to look at these kinetic constants a little bit. So we have these rate constants, K off and K on. And as I told the group earlier this morning, I really hope that this is taking you back to some happy memories of general chemistry if you looked at kinetics. And so in this case, K off is a first order rate constant. Why is it a first order rate constant? Because we have one species reacting, and that one species happens to be RL, the RL complex, and the RL complex decays to give us free receptor and free ligand. Because this is a first order reaction, our units are per second. Okay, now K on is the rate of the second, right, reaction under here. I should say it's the rate of the reverse reaction here, and it's a second order rate constant. Why is it a second order rate constant? Because we have two species that have got to combine, right? And how are they going to combine? They're going to find each other by random diffusion. So there's like a component here that's dependent upon their concentration, relative concentration. So the units here are per molar per second. And if we look at equation 1 here, right, the dissociation reaction, we have RL, which is a receptor ligand complex, degrading to give us free receptor and free ligand. And KD is a reduction of these two kinetic constants. It's the rate constant for the breakdown of the complex and the rate constant for formation of the complex. So the units for K off are per second, and the units for K on are per molar per second, because we have a concentration dependence in that second order rate constant. If we look at this ratio of the units, so this ratio down here for K off over K on, per molar per second. Per seconds cancel out. We're left with per molar on the bottom of this expression. And if we make this more palatable to our human eyes, we're going to end up with the concentrations of molar, right? So flip. So KD, the units of KD are expressed in terms of molarity. All right, now, if we look at this expression for Kd, we can express it in two ways. We can express Kd as a ratio of rate constants. Unless you're going to have some fancy equipment and some excellent detection systems, you can't determine these rate constants. It's crazy to do this. So thankfully, we can use this other expression for Kd. And so let me rewrite this because it's getting very messy. So we can express Kd in terms of the concentration of receptor free times the concentration of ligand free divided by receptor ligand. You know what? This is easier to use, right? This is what we use most times in the lab. Okay, now receptor free. Well, we don't know this. We have a hard time in some systems even determining how much receptor is there, right? So we can't easily determine R free in experimental system. L free, yeah, we know this. We know L free. How do we know? Because we added it. We took a pipetter and we added it, or we're able to measure it, right? RL, we know. We can measure. And so, you know, there's a way that we can change this equation around so that we can get experimental data that we can use to calculate Kd. And I am not going to take the time to derive that right now, but I want to show you the equation so that you can use it for your homework. So we can't know free receptor experimentally, not all the time. Sometimes you can, right? Like for the myoglobin system, you can because it's a colored protein, right? It has a great absorbance that we could use. So what can we measure? What can we know? We know L total because we add L total to the experiment. We know L total and we assume L free to be equal to L total because L free is so much greater concentration than R total. All right, or right, think about it as this, how I wrote it before. R total is very, very much less than L free in the system. So, right. Anyway, sometimes we can measure or know RL, right? So we can know this through absorbance. We can know this through, if we do, radio label studies, we can know the concentration of RL. I'm not going to do this now. Not now, interest of time. But we need to get this expression in terms of things that we can know or fit. But anyway, these are the equations you need for homework.